to thank the organisers for giving me this opportunity to present some research from my lab. It's been a really great meeting. There's been loads of talks by very talented scientists on the subject of talent. I found myself earlier on thinking about some of the most talented footballers in the world. I thought of Harry Kane, Phil Foden, Jude Bellingham, Alexander-Arnold, Bukayo Saka, and I was just feeling grateful that I'm not Denmark, basically. So, GABAA receptors are involved in loads of different things, sleep, and because they're involved in so many things, it makes them good targets for treating all kinds of disorders. You've got sleep, insomnia, they're involved in reward pathways, that's addiction, fear processing, that's things like anxiety, cognition, so maybe learning disorders, nociception, pain, excitation, inhibition, balance, that's like autism and epilepsy. Also things outside the CNS, sugar control, diabetes. So the holy grail really is to make a drug or a cocktail of drugs that can target across all the different subtypes in exactly the right way, so that you could basically not just treat all of these disorders, but you could fundamentally enhance brain function, and you could actually make the human brain better than it is now. And if you could do that, then obviously you can make a type of superhuman. I call them GABA woman or GABA man. And they would have various special properties. So they would be intellectually enhanced, they'd never get stressed, they'd feel no pain, they could eat as much candy as they wanted without ever getting diabetes. They'd always sleep really well like a baby, although not my baby, unfortunately. And then the final one, which is a little bit controversial, is that they could get high on recreational drugs as often as they wanted without ever getting addicted. So, there's some serious, potential in the future to be optimistic and happy. So there's drugs out there actually that target GABAA receptors and can do some of these things. There's, for example, diazepam, Valium. This is a benzodiazepine. There's loads of different types of benzos. There's also Z drugs, which are used to treat insomnia. And they all bind the pocket between the alpha and the gamma subunit that you can see over here. And so normally GABA receptors then, they're normally pentamers. You've got 2 betas, 2 alphas, and a gamma, and the benzos bind at the alpha-gamma pocket. And all the clinical drugs, they tend to enhance the function of the receptor. So you can't see it very well here, but before diazepam and then after diazepam, and if we zoom in, then you can see that there is a slight increase in the size of the inhibitory current. So there's a slight, a boost to the inhibition. And this is how these drugs kind of work. And we have some structures bound by benzodiazepines, diazepam, for example, and also there's some out there bound by a Z drug. But there's certain things, there's still quite a lot of things we don't really know about how they work at the molecular level. We don't really have examples. There are drugs out there that are selective, but we don't have explanations specifically for why they are selective molecularly. And we also don't understand the overarching transduction mechanism. If you look at this figure that I've got here, you can see the benzo bound, but there's 2 structures there that are overlaid. One is without the benzo, one is with. And when the benzo binds, pretty much nothing happens. So it's very hard to understand, how does it cause a conformational change that makes the receptor open more often? And this has led to the kind of this mysterious molecular glue hypothesis, where basically just somehow by the benzo binding and increasing the number of interactions, somehow that's changing the dynamics of the receptor. But obviously this is a very kind of hazy kind of explanation. We don't really know what's going on. So, there's also another thing, which is that we don't really have any structures with analog series of drugs that are all similar, where they transition from a negative to a positive allosteric transition. That might help us to understand a bit more, about what drugs do in this pocket to have different properties and to understand their design a bit better. So I'm going to look at 1 and 3 today in this story that I'm presenting from our recent paper. So we looked at alpha-5 then, and the reason that we looked at alpha-5 was because we were collaborating with Roche, and they have lots of really nice alpha-5 ligands, and these ligands have subtype selectivity, And they also show a transition from inhibitors to activators, or positive modulators and negative modulators. And alpha-5 also has been shown to be involved specifically in cognition. So in this example here, you can take, for example, a mouse, you put it in this cloudy water and let it find a hidden platform. And each time that it repeats the trial, it gets a little bit faster at finding the platform. This is the wild type animal. If you knock out alpha-5, then actually the animal gets better. It gets faster at finding the platform, and it learns more quickly. And so therefore, obviously, these drugs, are of interest to Roche. If you can make inhibitors, then you can actually boost learning. And if you make positive modulators, which they were also interested in, then maybe these can have some useful roles as well. And so for me, when I was thinking about this, it was it's just a bit of a weird thing. I mean, why would we have a gene that makes us learn, sort of impairs our ability to learn? It's not, I don't think, has ever been really solved. And it also raised other research questions to me, like it made me wonder if Donald Trump has really high expression levels of the alpha-5 subunit. I won't be answering that question, sadly, in this talk. Okay, so we've got the alpha-beta-gamma receptor then. There are no structures, actually, of... so there's structures, cryo-EM structures of the alpha-1, but there's no structures of alpha-5 heteromeric receptors. And at the time when we started this work, we were still trying things with crystallization and stuff like that. So I made this kind of chimeric construct where I basically got 5 alphas to form a pentamer, and we added in the benzodiazepine pocket on the other side. So this is specifically for alpha-5 that we were looking at. Lots of mutations involved. I'm not going to go through everything that we did. So obviously, a bit of a disclaimer then, this protein doesn't actually work as an ion channel because it doesn't respond to GABA, the agonist, so it can't open in response to anything. If you put it in membranes and you spray on benzodiazepine, then it doesn't actually open, which is not very surprising, really. But given that the pocket doesn't really undergo any obvious conformational changes with or without benzo, we're not really losing that information. We just want a structural template that we can use to understand better how these drugs bind, why are they selective, what's the difference between a positive and a negative modulator in these pockets in a related series of analogues. So hopefully it's a good template for binding. We solved some structures with sort of positive control drugs, so Valium, for example. We can see here's the density, so we can get to about 3 angstroms and fit the Valium with good confidence. If we compare it to previous alpha-1 structure with Valium bound, then we can see that they're pretty close. Same binding mode, fairly closely aligned. There's a little bit of difference. That difference might just come from the fact that ours is alpha-5, theirs is alpha-1. If we look at this cryo micrograph here, you can see zoomed in, you can see quite a lot of circles, hopefully. You can just about pick them out. So they're top views and not many side views. And what that means is in cryo-EM, you want to get an array of different views in order to build a good structure. And so because there aren't many side views, we have to collect more data. Still not too bad, but maybe we need a day of data or half a day of data to get about 3 angstroms. So it's not ideal, but that's what we worked with in the paper. There was also a demonstration of a megabody. So a megabody is something that can bind to a receptor. It's like a nanobody, but it's got an extra bit on it to make it bigger, like a type of antibody. And in this paper, they looked actually at this GABA receptor as well. They looked at a beta-3 homomer. Again, you can see loads of top views. And because they pretty much in this one, unlike ours, they had pretty much only top views, they couldn't build a good map. But if they used a megabody which bound to the receptor, then they suddenly got good tumbling in the ice, and they got a good range of orientations, top and side, and then they could get themselves a decent map of the protein. So just as an example, we tested this. We didn't do this for all of our structures, but we tested it on our sample. In our case, we used a different megabody, one that just binds to nanodiscs. So any membrane protein that you put in a nanodisc, you could use this megabody on. And instead of having mainly top views, you can see the odd top view, but we've got a whole raft around here. You probably can't see them very well, but lots of side views. And so that means that we could get better resolution. So in just 2 hours, you can get about 2.5 angstrom resolution. This is with a type of benzodiazepine bound, a different one from the Valium that I just mentioned. And if we do a day collection, then we can improve the resolution further. So you can see it's a bit more angular, but there's also a lot of extra information in this map that you can extract by doing different types of processing. So you can see here now we can actually see the rings of the benzodiazepine and the little side chains coming out, and we can also see the rings of the amino acids that are interacting with the drug. So, and we can actually see, if we look at this map compared to our original one, that in our publication we actually built this threonine wrong. We weren't sure how it was positioned, but you can see here actually it should be perpendicular, sideways, going in like this. So that, you know, these are small details, but they're useful if you're really wanting to do high quality drug design, really understand how drugs bind into a pocket. You know, it's not the kind of thing that's going to get people on the street excited, but if you're a computational chemist or a medicinal chemist, then these fine details, they're the kinds of things that are going to get you really super horny. So we've got here two drugs, flumazenil, and this one here, this row 4513. And the one that we solved in our pocket was this one. This one's been solved before. And the only difference between these two drugs, so functionally one's neutral and one's a negative modulator. The only difference is this azide over here. So logically you'd think at that resolution we could see that azide. Presumably it must be doing something, right? It must be interacting with something that's going to tell us why it's a negative modulator or not. But if we look at the map, actually this azide is just unresolved. So it's either floating around, just rotating freely in space, or maybe the negative charge is somehow affecting the cryo-M, because cryo-M is about electrons, which are negative. Sometimes you don't see negative side chains as much. So we don't get answers from these structures at this resolution for this particular molecule, showing that structure cannot always tell you everything you need. We also looked at an alpha-5 series of molecules from Roche. And so these ones are all related then. They've all got an upper half, which is this rigid two rings here, with an isoxazole, and then the rings vary a little bit. One's got no nitrogens, one's got one, one's got two nitrogens in the ring. And then you can see the lower part of the molecule is more variable between them. And all three of these drugs from Roche, they are all selective for the alpha-5 receptor. This is radioligand binding data that Roche did. And you can see, compared to alpha-1 versus alpha-5, they all have between 30 and 300-fold selectivity. So we could solve structures of these. You can see the maps down here and the molecules up here. And they all show the similar kind of thing with the upper moiety sort of along here, and then they've got a lower part going down, further down into the pocket. And you see there's a slight difference over here, which is just where the density is much better in this linker for this particular molecule, because it's a shorter linker. There's only the one carbon versus the others where you've got two atoms. So there's more flexibility, and so it tends to disappear. I can see if I can get this to play. I don't know if it doesn't come up. Can't see anything on here. Oh, there it is. Yeah. So this is just kind of showing you a bit more, you know, where the drug is bound, just for context. So you can see that the drug is in the extracellular domain in a pocket at the interface between the two subunits. And you can see it's stacked in this aromatic cage, stacking between aromatic residues. And this is very characteristic of drugs that bind to pentameric ligand-gated ion channels. So by looking at understanding how these drugs bind, we could understand their selectivity. So alpha-5 has two residues that are specific to it and that are different from in all the other alpha subtypes of receptors. So they've got a threonine here, which is a serine in all the other residues. So we've got an extra methyl group, which is capable of forming interactions. And it's also got an isoleucine here, this 215, which again is a valine in all the other subtypes. So it's got, again, another extra methyl. So alpha-5 has got two extra methyls that can form, and we can see they're both quite close to the molecule. They can form extra contacts, extra van der Waals forces, to help these molecules have higher affinity for the alpha-5 subunit. And we could do, so Roche did a load of radioligand binding work, and if you mutate this threonine to a serine, then you can see that the affinity for all three of these compounds drops quite significantly. And if you take alpha-1 and you change the serine to a threonine, then you get a recovery, you get at least some recovery of the binding affinity. Again, with the isoleucine, you get a reduction in affinity if you change it to the alpha-1 residue, and vice versa. If you put it in alpha-1, then you tend to get some recovery of affinity. And you can probably see from this as well that the threonine residue seems to have a bigger effect, is probably more important than the isoleucine, and that kind of makes sense because the threonine side chain is a bit closer to the molecule. So we can explain then with this series of molecules quite nicely some molecular basis of selectivity, which hasn't really been done before for GABA receptors. But these molecules, they also have different properties. So this is their percentage change in GABA response, GABA currents, some EPHIS data from Roche. And you can see that the one on the left, this basmizanil compound, is a negative modulator, and they've been trying this out, for example, in clinical trials to treat cognitive impairment associated with schizophrenia. And they've also been interested in positive modulators for various reasons. And they've got a couple of those here, which you can see enhance the current. So you've got, what is the reason why, what's going on? Is there any differences in the pocket that we can put down to the changes in modulation? Is there any difference in the kind of the binding modes of the drugs? Is there a transition that happens? And so we focused mainly on the upper part of the molecule because it's more similar between the three molecules, so it's easier to make conclusions about it. It's also nice and rigid, so we can be more confident about the changes that are happening. The lower parts of the molecule are a bit harder to map and be absolutely certain what's going on. But what we can see in the upper half of the molecule is that compared to the basmizanil, this positive allosteric modulator version has slid across the pocket by a small amount, a bit over an angstrom. Again, if we look at the other molecule, this has also slid across the pocket. The change is a little bit greater. So in the positive drugs then, there tends to be a bit of movement, a bit of motion in the upper half of the molecule across the pocket. And if we think about the molecular contacts that are involved, we can see that this ring in the negative modulator is stacking underneath this tyrosine ring, and that brings it fairly close to these two residues over here, this phenylalanine and this histidine. If we look at a positive modulator, then because it's got this nitrogen, it seems to move away a little bit from the tyrosine so that the nitrogen can form an interaction, a polar interaction with the oxygen or the hydroxyl on the tyrosine. And so its distance away from this histidine and this phenylalanine grows a bit. And again, if we look at the other molecule, it's got two nitrogens, and now it seems to shift a little bit further along still to form polar interactions with these two residues over here. And again, the distance to these two residues, phenylalanine and histidine, grows. Now, these changes are quite small, especially when we're thinking about the resolution of our maps here, which are about 2.7 to 3 angstroms. So we can't be 100% confident this is definitely happening, but we did mutagenesis work. Roche did explored some mutants, and they did radioligand binding. This was all using wild type alpha, beta, gamma, alpha 5 receptors. And if you mutated the phenylalanine residue, then in the situation where it's closer and it's involved in binding, if you get rid of it and replace it with an alanine, then you get a big impact on binding, 20-fold. The same for this other drug, but not for the one where it's further away and it's not involved in binding anymore. It doesn't matter if you mutate it, it doesn't have any impact. And we showed the same thing again for the histidines, that for the negative modulator, if you mutated it to an alanine, it had a significant effect. For the other two, it didn't do so much. So again, the mutagenesis data supports the idea in wild-type receptors that these drugs are sliding along and moving away from these two residues. So this suggests then that the motion, the displacement in the upper half of this molecule within the pocket could contribute to the modulation, whether it's negative or positive. But obviously we've also got a lower part of the molecule that we've not been considering here, and it could be that that is just doing all the work in terms of changing the function, and the upper part is just moving around as a consequence of this. So Roche provided a hybrid molecule. In this hybrid molecule, we've got the upper part of the positive modulator and the lower part of the negative modulator. And when you look at the EPHIS from this molecule, you get something in between. So what this tells us is that each half of the molecule is contributing roughly half of the effect. And when we solve the structure of this molecule, again, we could see this transition, this movement of the upper half, which is contributing to the PAM effect, but the lower half still mapped over the original negative modulator. And if we compare the upper half where it's gone to, then we can see it fits very closely over our previous positive allosteric modulator. So what this tells us then is that the upper half, by sliding around, does contribute to the transition from a negative to a positive modulator. It's contributing to about half the effect. And it also tells us that these drugs, these kinds of drugs where you've got these two segments via a linker, you can use them in a kind of plug and play capacity where they're two independent modules. You can tweak them and change them to contribute to the negative or the positive effect. So this is interesting from a drug design perspective. Maybe these types of molecules where you've got an upper and a lower part could also be used in other subtypes to control the amount of modulation that you can get in the pocket. So we still can't say what they do to drive transductional effects, but we can say here that we've got a series of molecules, and it seems that their positional displacement in the pocket affects whether they're positive or negative, and they can work as little plug and play modules. So just going to stop there and just acknowledge my lab, especially Vikram, who's involved in doing quite a bit of this work, the cryo-EM work and building the models. And also collaborators at Roche, Maria Clemencia Hernandez and Giuseppe Cesare, who were involved in making the molecules, doing all the leading the radioligand binding work, making sure that happened, and funders. And that's everything. Thank you.
Any questions for Paul on a lovely talk?
Thanks. Beautiful structures. Considering how high the resolution is, the compound that had the azide and you couldn't see the azide, is there any chance at all that it got reduced at some point during the sample preparation?
I guess that is a risk, but normally, that molecule is alcohol antagonist is normally pretty stable. So, you can keep that in solution and do functional studies, 1 month to the next. It still maintains its function, so I would hope.
You don't need reducing agents during your protein prep for cryo-EM, for example?
No, we don't do it. It's very standard buffer, so we would add the drug right at the end. There'd just be a bit of salt and some HEPs, nothing else. So.
Hey, Paul, can you hear me? Nice talk, congrats on the paper. If you could do, because I guess Ross didn't do single channel recordings, right?
No.
But if you did the single channel recordings, you get a positive or negative allosteric modulator, you could imagine that increases the open probability or reduces it, or enhances desensitization, or reduces desensitization. So if you could do the experiments, what do you think you'd get?
In terms of single channels?
Because no one's ever really nailed that for benzos, how they work.
No,
Right.
the main...
What did they actually do? What did they do with the channel, right?
Yeah, so that is the big mystery. Obviously, the structures don't really help us much. They just tell us the binding mode. But
if you had to guess, what do you think it would be?
Well, I guess the main study, which was that I'm familiar with from Trevor Smart's lab, was really that they came up with a proposal that it increases the affinity so the benzo binding will increase the affinity of the second GABA binding.
Right.
So you will get an increase in probability of open and closing, but you shouldn't, in theory, get a shift to desensitization because it's an affinity-based situation.
Yeah, we can chat about it later, but I guess it's assuming that when binding occurs, there's energy from that. But another way to think about it is when they bind, they stabilize ongoing conformations in the protein itself, and that isn't driving anything, it's stabilizing existing structures. It's a bit like kind of 20 Auerbach's kind of thermodynamic way to think about it.
Yeah, exactly, yeah.
And it'd be interesting to see what you'd get now that you have this series and you have the structures of it.
Yeah,
Anyway, congrats.
think we've got time for maybe one more quick one if there's a question out there. Okay, we'll thank Dr. Paul Miller